ELISA, enzyme-linked immunosorbent assay, is a highly sensitive method for detecting specific antigens or antibodies in a sample. It can be performed in different ways, direct and indirect. In direct ELISA, an enzyme-labeled antibody directly detects the antigen or antibody of interest. In indirect ELISA, a primary antibody binds to the sample's antigen-antibody complex, and a secondary labeled antibody detects the primary antibody. The steps in an indirect ELISA for detecting an antigen or antibody are as follows. Initially, we prepare the sample by putting antigens or antibodies in a special liquid. Then we stick a special antibody to a solid surface. After that, we add the sample's antigen, which sticks to the special antibody. Then we add another special antibody, which can detect the first one. This second antibody has a label. Finally, we add a substance that changes color when the label is present. The color change shows how much antigen is there. We can see the results by looking at the color or using a machine. ELISA is used in medicine and research because it's very sensitive and measures things accurately to find specific antigens or antibodies in samples. In competitive ELISA, both an enzyme-linked control antigen and a sample containing an unknown quantity of unlabeled antigen are added to a sample well coated with antibody. In this assay, the sample is added first and the bound antibody captures the unlabeled antigen. The labeled antigen is then added, and a reversible equilibrium is established between the amount of labeled control antigen and sample antigen bound to the antibody depending on their relative concentrations. If the ratio of labeled control antigen to sample antigen is high, then the signal will be maximized. If the ratio of labeled control antigen to sample antigen is low, then the signal will be weak. Because the standard curve in a competitive ELISA exhibits the maximum signal at the lowest concentrations of sample antigen, this assay is very sensitive. These figures show the types of ELISA mentioned before. Notice the differences in structures for different types of ELISA. Various immunoassay test kits based on the competitive ELISA are commercially available for measurement of organic contaminants such as pesticides, petroleum and PCBs in environmental samples. These kits are approved for use by the US EPA for detection and monitoring of these contaminants. Several groups have also worked on development of monoclonal antibodies for detection of various metals. Metal detection is problematic due to the small size of the metal atom, which does not elicit an immune response, and so antibodies are not produced. This problem has been overcome by using a metal chelator complex to produce specific monoclonal antibodies. Commercial kits use various methods to capture contaminant antigens, offering sensitivity comparable to traditional approaches for detecting organic contaminants like pesticides and PCBs. These kits provide detection limits from high parts per billion to low parts per million. In a 1996 study comparing a commercial immunoassay kit to GCMS analysis for 161 samples from a PCB-contaminated site, the immunoassay had a 3.1% false positive rate and an 11.2% false negative rate. Competitive ELISA is good at finding really tiny amounts of a substance, but sometimes it's so good that we have to mix the sample a lot to make sure we're right. It's also quick, easy, and can be used in faraway places, and it's cheaper than some other methods. The main issue with tests like this is they might react to things that look a bit like the substance we're looking for. For example, if we're checking for a specific kind of pollutant, the test might react to other similar pollutants, making it seem like there's more of the pollutant than there really is. Magnetic immunoseparation is a method that uses tiny magnetic beads coated with special antibodies to grab and collect specific substances, like microorganisms, proteins, or DNA. It's a bit like using a magnet to pick up metal objects. In this process, the antibodies on the magnetic beads stick to the substances we want to collect. Then, we use a magnet to pull the beads out of the solution, leaving the unwanted stuff behind. These magnetic beads are really small, about 75 nanometers to 20 micrometers in size, and they're usually made of a material called magnetite. They respond strongly to magnets but don't stay magnetic when the magnetic field is gone. This method is handy for isolating and purifying specific substances in a controlled and efficient way. This is a schematic representation of the principle of immunomagnetic separation. In the first tube is a solution containing the antigen such as virus. Antibodies specific to the virus are added to this solution. These antibodies, which have been attached chemically to a paramagnetic particle such as magnetite, then bind the virus. When a magnet is applied to the solution, the magnetic particle is attracted to the magnet along with the attached antigen. Immunomagnetic separation is a valuable tool for concentrating and purifying antigens from complex solutions. Immunomagnetic separation has several advantages. It is good at separating the target substance efficiently, it's cost-effective, and it can be automated, making the process easier. It is often one of the simplest methods for isolating specific substances. However, there can be issues with the method reacting to things it shouldn't, like other similar substances. But usually, we can find ways to fix that. Just like with any microbiology process, immunomagnetic separations need to be optimized and done in the right way, like choosing the right method, whether it's using a microcentrifuge or a column matrix.